So today we're going to be looking at an evaluation of perigold vesicles containing retrograde but not anterograde proteins consistent with the cisternal progression model of the intra-Golgi transport. This paper is by Martinez-Menarguez et al. And let's start. Okay, so first we have our overview and importance of this paper. The Golgi complex plays an important role in the secretory pathway. It is the central organelle for newly synthesized proteins. They travel through it to get to their final destination. Two models have been created in order to explain this movement. That's the vesicle shuttle model and the cisternal maturation model, which is also known as the cisternal progression model. The vesicle shuttle model predicts that COP1 coded perigold vesicles contain anterograde transported cargo. This model shows small vesicles that bud from the Golgi cisterna using COP1 coat and mediate forward transport throughout the Golgi. Each cisternae in this model has a specific set of resident enzymes. However, in the cisternal maturation model, it predicts that perigold vesicles contain Golgi resident proteins that are more retrograde in movement. This model shows that endoplasmic reticulum-derived membranes are imported at the cis side of the Golgi, while vesicles are formed at the trans side. This results in the progression of each entire cisternae from the cis end to the trans end. And this means that enzymes must be transported backward from a trans-moving cisterna to a more cis-positioned cisterna. This study focuses on differentiating between perigolgi vesicles and other vesicles in the Golgi region in order to discover the true role of perigolgi vesicles in the movement through the Golgi complex. Okay, and so some of the methods that they used in this paper were antibodies, GFP transfection and trafficking, ultra-thin cryosectioning, immunogold labeled cryosections, and quantitative IEM. Within the quantitative part, there was cis to trans and lateral distribution of Golgi-associated proteins, density labeling of MAN2, VSVG, and giantin, and double immunogold analysis. And then the cells that they chose for this specific study were NRK cells, and they were used because of the presence of Golgi stacks observed in a pre-immunogold labeling of many different types of tissue. Okay, so let's look at the results. So this is figures 1, 2, and 3. One of the prominent discoveries from this paper was that mannosides 2, or MAN2, is present in perigolgi vesicles. MAN2 is a Golgi resident enzyme with a short transmembrane domain and a luminar catalytic domain. It's involved in end-linked glycosylation. In immunogold labeling, it was found that the majority of this MAN2 enzyme was present in cisternae G2, which is in more cis position, while lower amounts were found in G1, G3, G4, and G5 had almost none of the enzyme present. In figure 1 here, this shows the immunogold labeling of MAN2 on ultra-thin cryosections of NRK cells, showing the presence of MAN2 in the cis medial Golgi cisternae at spot G, which is right here, and in the lateral rims of the cisternae and associated vesicular profiles, which are shown by the arrows. The distribution of MAN2 between cisternae and associated vesicles varies between the Golgis. The arrowhead near A, right here, points to a MAN2 positive Golgi lateral rim with a clearly visible COP coat, and so does C. In figure 2, this is showing MAN2. They're both labeled by different sized gold particles. So the MAN2 is labeled with the larger gold particles, and then the COP2 is labeled with smaller gold particles, and it shows the relationship between the MAN2 and the COP2. It's showing that these vesicles, the perigolgi vesicles, lack COP2, and you can kind of tell in this that the COP2, which are down here near the ER, they are restricted to vesicles close to the ER, showing that it's not very present in the majority of the Golgi complex. In figure 3 here, we're looking at COP2 co-localizing with MAN2 in lateral rims, shown by the arrowhead down here. The perigolgi vesicles are shown by arrows, and COP1 labeling is more widely dispersed, present on the perigolgi vesicles and at the Golgi cisterna. Quantitative analysis data from these figures showed that about 20% of the MAN2 positive peri-Golgi vesicles were labeled for COP1, but only 2% for COP2. This defines the COP-coded cisternal buds and all coded peri-Golgi vesicles as the COP1 type. Okay, so for figure 4 here, GFP transfection was used to directly compare the Golgi distribution of MAN2 with VSVG detected by immunolabeling of cryosections. This figure is showing VSVG tagged with GFP present in Golgi cisternae of transfected NRK cells, but not in lateral peri-Golgi vesicles. A right here shows that after 2.5 minutes at the permissive temperature, VSVG co-localizes with the K-del-R in BTCs, but is absent from the Golgi complex. Also, K-del-R is present in Golgi cisternae and in surrounding vesicles shown by the arrows. In B, we are seeing after 20 minutes of the temperature block, VSVG is present in all cisternae of the Golgi complex, and K-del-R positive peri-Golgi vesicles do not contain VSVG, which is again shown by the arrows. In C here, we're looking at an example of a cell with high expression levels of VSVG, GFP, and based on these observations, it was concluded that MAN2 distribution over the Golgi complex is not affected by VSVG, which in this case was tagged with GFP. Okay, so this shows figures 5, 6, and 7. They show the comparison of labeling patterns between MAN2 and VSVG in relation to other Golgi associating proteins such as giantin, K-del-R, and RBET1. So in figure 5 here, it's showing giantin and RBET1. Giantin showed a consistent labeling pattern with a majority of labeling in the Golgi complex and associated vesicles. It was also found in COP1-coated membranes and rims with relatively the same quantitative trend as MAN1 in the earlier results. RBET1 is a snare protein, and it labels BTC membranes and the cis-Golgi cisterna. When it is doubled with giantin, about 60% was located in COP1-coated buds and peri-Golgi vesicles. Figure 6 shows MAN2 co-localized with giantin and K-del-R in different populations of peri-Golgi vesicles. A shows MAN2 co-localized with giantin in peri-Golgi vesicles, which is shown by the arrows. B shows that MAN2 is regularly found with K-del-R in peri-Golgi vesicles, which again are shown by the arrows. And C, despite the high labeling densities of both the K-del-R and giantin, they are mostly found in different populations of peri-Golgi vesicles, which is seen here. Okay, so in figure 7, this shows K-del and RBET co-localized in peripheral VTCs, but not peri-Golgi vesicles. These figures in the data collected all showed that the high percentages of giantin, K-dels, and RBET1 are present in Golgi-associated COP1 membranes and lateral rims. Also that MAN2 resembles the same trend along each of these Golgi-associated proteins. And on the other hand, enterograde protein, VSVG, shows a completely different trend, which dissociates it from the COP1 membranes. Okay, so to wrap up the discussion of this paper, this study demonstrated that NRK cells with specific amounts of enzyme MAN2 localized to COP1-coated peri-Golgi vesicles. This observation satisfies the critical requirement for the cisternal progression model. The study also found that VTC's secretory proteins are excluded from recycling COP1-coated membranes compared with recycling proteins such as K-del-R and RBET1. This led to the hypothesis that enterograde cargo is concentrated in VTC tubules because COP1-coated vesicles without cargo detach from them. It also has identified different classes of peri-Golgi vesicles, determining that COP1-coated peri-Golgi vesicles are most likely involved in retrograde and not enterograde transport through the Golgi complex. Some other evidence before I go that peri-Golgi vesicles contain retrograde proteins are that MAN2 and giantin concentrations are the same in peri-Golgi vesicles and the Golgi stack, while VSVG concentrations are much lower. Also, MAN2 localizes with K-del and giantin, which indicates the existence of two types of MAN2 peri-Golgi vesicles. Thank you.